Hello students. In this lecture, we will discuss two important eukaryotic cell organelles, the mitochondria and the chloroplast. Let us begin with mitochondria. Mitochondria play a critical role in aerobic respiration for the generation of metabolic energy in the form of ATP. Adenosine triphosphate, and hence, are referred to as powerhouses of the cell. Structurally, a mitochondrion is a double membranous cell organelle consisting of outer and inner mitochondrial membranes, separated by an intermembrane space. The outer membrane is smooth and porous, whereas the inner membrane is thrown into numerous folds, called cristae. Present on the cristae are stalk-like structures, called the F0, F1 particles, also termed ATP syntheses. These are directly involved in the synthesis of ATP. The mitochondrial membranes enclose a fluid matrix that accommodates the circular DNA, 70S ribosomes, and the enzymes required for the process of aerobic respiration. Though mitochondrion has its own genetic material, it is still dependent on the nuclear DNA for certain functions. This makes the mitochondrion a semi-autonomous cell organelle. Functionally, the mitochondria are involved in the aerobic respiration process. During aerobic respiration, glucose is oxidized to generate metabolic energy in the form of ATP. The process of aerobic respiration is a sequence of three events, namely, the glycolysis, the Krebs cycle, and the electron transport chain. Of these three events, the Krebs cycle and the electron transport chain occur in the mitochondria. Let us learn in brief about aerobic respiration. The first step of aerobic respiration is glycolysis. Glycolysis occurs in the cytosol of the cell, where glucose is oxidized to form two molecules of pyruvate. The formed pyruvate is transported to mitochondria and is converted into acetyl-CoA by a process called oxidative decarboxylation. Acetyl-CoA enters the Krebs cycle that takes place in the matrix of the mitochondrion. Here, the acetyl-CoA is broken down into carbon dioxide and water, yielding NADH and FADH2 as byproducts. The formed NADH and FADH2 reach the inner mitochondrial membrane to enter the electron transport chain to finally yield ATP. Each glucose molecule, upon complete oxidation by aerobic respiration, yields 36 to 38 ATP molecules. These are the functional details of mitochondria. Before concluding mitochondria, let us understand an important concept called maternal inheritance. The genetic material of mitochondria can be inherited from a parent to the offspring, but always from the female parent only. This makes the mitochondrial DNA maternally inherited DNA, and the type of inheritance is called maternal inheritance. There are also certain disorders associated with mitochondrial defects. When mitochondria fail to function, or if there are any defects in mitochondrial DNA, they are often associated with diseases such as LHON, Leber hereditary optic neuropathy, myopathy, etc. These are the characteristics of mitochondria. Let us now move to the next cell organelle, chloroplast. Chloroplasts are plastids present in plant cells that play a critical role in photosynthesis. In many aspects, chloroplasts are similar to mitochondria. Both organelles generate metabolic energy in the form of ATP, contain their own genetic material, and divide by fission. However, the chloroplasts perform a unique task called photosynthesis. Structurally, chloroplasts are double membranous cell organelles. The membranes enclose a fluid matrix called stroma, and membranous sacs, called thylakoids. Thylakoids are flattened discs and are arranged in stacks, called grana. There are many sets of grana present in the chloroplast and are interconnected by stromal lamellae. Functionally, the chloroplast is involved in photosynthesis. Photosynthesis is a process by which plants transform light energy into chemical energy. The energy from sunlight is harvested and used to drive the synthesis of glucose from carbon dioxide and water in the presence of the photosynthetic pigment, chlorophyll. In photosynthesis, 6 molecules of carbon dioxide and 6 water molecules are utilized to generate 1 molecule of glucose and 6 molecules of oxygen. The entire photosynthetic reaction can be split into 2 reactions, namely, the light reaction and the dark reaction. The light reaction occurs in the thylakoids and is hence called thylakoid reaction, during which sunlight is utilized to split water to generate oxygen, ATP, and NADPH. The generated ATP and NADPH are utilized by dark reactions to reduce carbon dioxide to glucose. These are the structural and functional details of the chloroplast. Apart from chloroplast, there are 2 types of plastids present in the plant cell, namely, the chromoplasts and leucoplasts. Before concluding, let us understand a theory called an endosymbiotic theory. This theory states that the eukaryotic cell organelles, the mitochondria, and chloroplast have evolved from prokaryotic cells. The similarities between the prokaryotic cells, mitochondria, and chloroplasts, with respect to their DNA, ribosomes, and mode of division, stand as support to this theory. These are the details of the mitochondria and chloroplasts.
Let us summarize.
Mitochondria and chloroplast are the double-membranous eukaryotic cell organelles. Mitochondria are double-membranous cell organelles consisting of outer and inner mitochondrial membranes, separated by an intermembrane space. Mitochondria possess their own genetic material. Mitochondria are referred to as semi-autonomous cell organelles. Mitochondria are involved in aerobic respiration to generate metabolic energy in the form of ATP. Chloroplast is a double-membranous plant cell organelle enclosing thylakoids and stroma. Chloroplast is involved in photosynthesis. The light reaction occurs in thylakoids, and the dark reaction occurs in the stroma. Endosymbiotic theory states that the mitochondria and chloroplast have evolved from prokaryotic cells.